Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I'm Dr. Shahid Amin, lecturer, Department of Chemistry, Quad University of Science and Technology. This is the first part of fifth lecture on biosynthesis of natural products. The course code is Chem 524, and in this part of this lecture, we'll be discussing about the building blocks for secondary metabolism. So the topics that we'll be discussing in this part, these include primary metabolism, just an introduction, then introduction to secondary metabolism, a brief introduction, and building blocks for secondary metabolites. The reference book that the students will be following, this is Medicinal Natural Products by Paul M. Dweck, a biosynthetic approach, and this is third edition in 2009 by John Wiley and Sons. So first we look at primary metabolism. These are chemical transformations that are happening within the living organisms and that are responsible for running the business of life, whatever is needed for life, for example, energy and different materials that are inside the body. These are a result of the primary metabolism. And metabolism itself, this is an integrated network of enzyme-mediated and carefully regulated chemical reactions. And the pathways involved there, these are termed as metabolic pathways. For general continuity of life, two things are needed. One is energy, the second one, this is building blocks in order to make some higher molecules. And the general molecules that are present in the bodies of the living organisms are the molecules that the living organisms obtain from their environment as in the form of food. These include carbohydrates, proteins, fats, and nucleic acids. And these are the crucially important molecules that are called as the biomolecules, or the molecules of life. Organisms are very widely in their capacity to synthesize and transform chemicals. For example, plants are efficient at synthesizing organic compounds via photosynthesis. That is a peculiar process that is actually special for plants that are also called as the producers, because they can produce their molecules that are needed for their daily life themselves. But animals and microbes, they cannot produce their food themselves, so they have to rely upon plants for their food. Pathways for general modifying and synthesizing carbohydrates, proteins, fats, and nucleic acids, these are found to be essentially the same in all organisms, whether these are animals or plants or microbes. So although there may be small variations, there may be some differences, but these differences, these are minor differences, not the major ones. Otherwise, the metabolic pathways, these are general for all the organisms. And these processes demonstrate the fundamental unity of all the living matter. That is, all the living matter, this is based upon the same fundamental principles. And the chemical reaction that are that are taking place within the living organism, these are similar, and this type of metabolism is known as primary metabolism. And the metabolites are the chemical substances involved in this type of metabolism. These are known as the primary metabolites. Now we look at what we mean by secondary metabolism. Secondary metabolism or secondary metabolic pathways, these are concerned with compounds that have much more limited distribution in nature. So the compounds called secondary metabolites, these are found in only specific organisms or group of organisms, and these are actually the expression of individuality of the species. So not all the species or all the organisms, they will be producing the secondary metabolites. Rather, only specific organisms, they will be producing some specific type of secondary metabolites. Secondary metabolites are not necessarily produced under all conditions. And in the vast majority of the cases, the function of these compounds and their benefit to the organism, these are not well known yet in many cases. their exact function is not known. Out of these secondary metabolites, some are produced for easily appreciated reasons. For example, they may be toxic materials that may provide a defense mechanism against the predators, or they may be produce a volatile attractants towards same or the opposite species. In the same species, for attracting the opposite sex, some specific molecules are produced, and there may be that one organism that is producing some secondary metabolite that may be attracting some organism of the other species for some specific purpose. These may be the coloring agents that help the plants or some other organism to attract some other species. But it's logical to assume that all do play some vital role for the well-being of the producer that is producing these secondary metabolites. Now we look at the building blocks for secondary metabolites. So secondary metabolites are produced from molecules that are actually part of the primary metabolism. So secondary metabolites are actually derived from the primary metabolites. So out of the primary metabolism, if we talk about glycolysis process, that is actually the process of breakdown of glucose, that is a 6-carbon sugar, into simpler sugars or other molecules. So we'll start from this glycolysis. How this glycolysis gives us some primary metabolite that may result into formation of the secondary metabolism. For example, D-glucose, this enters glycolysis process. It gets phosphorylated to become glucose 6-phosphate, that is converted to glyceraldehyde 3-phosphate, and that is converted to 3-phosphoglyceric acid. Afterward, that is converted to phosphoenol pyruvate, and that is ultimately converted to the final product of glycolysis process that is known as pyruvic acid. Now, this glucose 6-phosphate that is formed as a result of the phosphorylation of glucose, this is changed to, in addition to being changed to glyceraldehyde 3-phosphate, in the glycolysis process, it also enters another pathway that is known as pentose phosphate cycle, and this is converted to erythrose 4-phosphate, that is a 4-carbon sugar. This molecule is also produced as a result of photosynthesis process in the green plants, and this is very important chemical substance with respect to its role in the biosynthesis of the secondary metabolites. For example, erythrose 4-phosphate in combination with phosphoenol pyruvate gives rise to a very important chemical substance known as a Shikimic Acid that results into formation that goes in towards the formation of amino acids like phenylalanine, tyrosine, and tryptophan. And all of these three amino acids, they are important substances with respect to the biosynthesis of the natural products. A lot of natural products like these what we call as the nitrogen containing secondary metabolites or alkaloids, these are produced from these amino acids. One of the intermediate in glycolysis process, that is 3-phosphoglyceric acid, it's also produced. In addition to being produced by glycolysis process, this is also produced during the process of photosynthesis. So this molecule gives rise to L-serine, another amino acid that gives further that is responsible for the formation of glycine and cysteine amino acids. The end product of glycolysis process, that is pyruvic acid, this gives rise to amino acids like valine, alanine, and leucine. This leucine is responsible for formation of a very large number of secondary metabolites. Pyruvic acid in combination with the glyceraldehyde 3-phosphate gives rise to deoxyxylulose 5-phosphate, that is actually a precursor of methyl erythritol 4-phosphate, and methyl erythritol 4-phosphate, this is an important precursor for the formation of secondary metabolites. Then pyruvic acid is converted to acetyl coenzyme A, that is itself a building block for secondary metabolites. It's converted to mevalonic acid, another important building block for secondary metabolites, or it may take part into Krebs cycle. And in the Krebs cycle, um, when it takes part and combines with oxaloacetic acid in order to give 2-oxoglutaric acid, that is converted to glutamic acid by a specific chemical reaction that we call as the transamination reactions. And this glutamic acid is further converted to another amino acid, ornithine. Ornithine is a amino acid that actually doesn't take part in the formation of the protein. Rather, this is non-proteinaceous or amino acid that doesn't there's no role in the formation of the proteins, but this has some other roles, and this goes into formation of arginine. Ornithine itself is responsible for the formation of different secondary metabolites, so it acts as a precursor for secondary metabolites. Similarly, oxaloacetic acid, that is product of that is an intermediate in Krebs cycle, or we can say this is a metabolite of Krebs cycle. It gives rise to aspartic acid, and aspartic acid further it gives rise to isoleucine, L-methionine, and L-lysine. L-methionine and L-lysine, both of these amino acids, they give rise to a large number of secondary metabolites. So they are acting as the precursors for secondary metabolite. So in this slide, the molecules that we have highlighted with this yellow color, these are actually the precursors towards secondary metabolite, although these are the primary metabolites, but they form secondary metabolites. Now we look at the primary metabolites that are providing us with some specific units or specific building blocks containing different number of carbons for this biosynthesis of the secondary metabolites. Amongst these, if you look at L-methionine, L-methionine contains a methyl group that is attached to the sulfur atom. This methyl group, actually, this is a source of 1 carbon units for a large number of secondary metabolites. And this 1 carbon unit, normally this is in the form of a methyl group, for example, for in the form of methoxy group, or in the form of the N-methyl groups in different molecules. And this methyl is normally attached with some other heteroatom, maybe oxygen, nitrogen, sulfur, or carbon. Like it may be oxygen, so in the form of methoxy, it may be nitrogen in the form of N-methyl, it may be sulfur, so S-methyl, or it may be simply another carbon atom. So C1 unit will be provided by L-methionine. Then C2 units, these are provided by 2 types of molecules. Actually, these are the major main primary source. This is acetyl coenzyme A. So both carbons of acetyl coenzyme A, of this acetyl part, these act as the C2 unit. And activated form of acetyl coenzyme A, this is malonyl coenzyme A, further activated, although this is also activated, but more activated form, this is malonyl coenzyme A, and this also provides us with 2 carbon units for the biosynthesis of secondary metabolites. Then we have a 5 carbon unit that is known as isoprene unit. Isoprene unit is produced from 2 types of molecules. One is acetyl coenzyme A, the other one is deoxyxyrillose phosphate. 3 molecules of acetyl coenzyme A combine in order to give rise to what we call as mevalonic acid, and this mevalonic acid was a very important molecule for the biosynthesis of terpenoids. The basic unit is isoprene unit. And the second one, that is deoxyxyrillose phosphate, that results in the formation of isoprene unit. This is through methyl erythritol phosphate. Then we have the amino acids like phenylalanine and tyrosine. Both of these amino acids, they provide us with the C6, C3 units. Out of this, building blocks, this C6, this is aromatic, and C3 is in the form of aliphatic chain. And this further could may be modified into C6C2 or C6C1, but the main source is C6C3 that is produced from these two amino acids. Phenylalanine and tyrosine, these are responsible. Tyrosine here we see this is producing C6C3. It also produces C6C2N, that is similar to this one, but one carbon is less as it's here. And to this unit there is attached one nitrogen atom, so they play a very important role in the biosynthesis of alkaloids. Then indole containing units, these are produced from tryptophan amino acid. So these units contain 2 more carbons as an aliphatic carbon along with 1 nitrogen. So this unit or this building block will be known as will be called as indole C2N block. Similarly, amino acid ornithine, this gives rise to C4N units. This is aliphatic unit with 1 nitrogen that may go towards formation of a ring and to produce this 5 carbon nitrogen containing 5 carbon heterocycle. L-lysine, it provides us with 6 carbon 6 member cyclic system with a nitrogen atom, so a heterocycle with a nitrogen atom. This is produced via C5N, that is aliphatic unit along with 1 nitrogen. So these are different building blocks that are actually produced from primary metabolites and are utilized in the formation of secondary metabolites. Now look at some real examples where we can identify different building blocks in the structures of these molecules, like orsilinic acid, that is a phenolic compound, and it contains 4 C2 units. These units are highlighted here as these bold lines. So 1, 2, 3, and 4 units are here. Similarly, another molecule that is parthenolide. Parthenolide contains 3 C5 units. C5 unit that we just discussed, that are isoprene units. We can identify these isoprene units here. This is 5 carbon unit that is highlighted as bold lines. Then again, this is another one, and this is another one. In case of naringine, we have a C6C3 unit that we can identify here. Then we have 3 C2 units that are here in the form of this phenolic compound, along with these 2 sugar molecule, glucose and rhamnose. In case of podophyllitoxin, we have 2 C6C3 units that we can identify here. These are these 2 units, along with 4 C1 units that are present in the form of these O-methoxymethyl groups, along with the CH2 group in between these 2 oxygen atoms in order to make a 5-member ring. Similarly, in case of tetrahydrocannabinolic acid, we have 6 C2 units that are we can recognize here, 1, 2, 3, 4, 5, and 6, along with 2 C5 units that are the isoprene units. 1 isoprene unit is here and the other one is here. Similarly, in case of this molecule that is papaverine, in papaverine we have 1 C6C2N unit that we can identify here. Then we have a C6C2 unit that is here. Actually, it's formed from C6C3 unit, along with 4 C1 units that are present in the form of these methyl groups of the methoxy. In this molecule, lysergic acid, we have indole C2N, that is, we can identify this is indole, and this is 1, 2, and this is nitrogen. So indole C2N, this is here. Then we have 1 C5 unit that is isoprene unit, along with one C1 unit in the form of this N-methyl group. Then in case of cocaine, we have C4N that is present here as this pyrrolidine ring. Then two C2 units we have here. Then C6C1 unit that is actually produced from C6C3 unit, and this we can identify here. And two C1 units that are in the form of, one is N-methyl and the other one is the methyl group of this ester. So these are some examples where we can identify the basic units that are actually making up these molecules as their building blocks.